Hello viewers, welcome to Mythology. In this video, we would be focusing on the overview of a cell cycle. The cell cycle is a series of cyclic events that take place in a cell while the cell divides into two daughter cells. These cyclic events include growth, checkpoints, and the divisional stages. It usually completes in two major steps, starting with a typical diploid cell. Cell cycle completes in two broad steps. It is interphase followed by an M phase. Interphase was earlier believed to be a resting stage, but later these were the most active stages undergoing thorough preparation for division. Actually, the DNA duplication, protein synthesis, and other such processes that occur during interphase, none of these can be seen from outside. So, the scientists must have thought it to be a resting stage. During interphase, there is cell growth, nutrient accumulation, DNA replication, transcription, and translation. There are also quality control checks called the DNA checks. This interphase is subdivided into three phases: G1 phase, synthetic phase, and G2 phase. After that, it follows the M phase, which is followed by cytokinesis. Beyond cytokinesis, the cell may either continue into the cell cycle or it may come out of the cell cycle depending upon the conditions prevailing there. Let's begin with the G1 of interphase. This is called as the growth phase 1. The cell size increases. The cellular content duplication except the chromosomes occur during this phase. There is mRNA synthesis, which is called transcription. There is also protein synthesis, which we term as translation. It also comes up with the synthesis of ATP. When everything is going good and no errors exist, then it moves to the next phase called synthetic phase of interphase. Now let's see what happens in the synthetic phase. During synthetic phase, there is synthesis of DNA. The deoxyribonucleic acid is duplicated here, or we can say that DNA replication takes place. The histone proteins are also synthesized in all eukaryotes. Other than that, the centriole also divides, but that is only in animal cells. The phase that follows the synthetic phase is the G2 phase. During G2 phase, which is called the growth phase 2, the cell rapidly grows again. Moreover, during the G2 phase, the tubulin proteins are synthesized, which is required for the formation of spindles. The ATP synthesis increases with an increase in the synthesis of RNA. These are preparation to enter into the M phase, that is the divisional stage. It is then The cell sinks into the M phase with karyokinesis followed by cytokinesis. With the onset of the M phase starts the prophase stage. The prophase, during which the chromatin condenses into chromosomes. The next is the metaphase, where chromosomes line up along the imaginary metaphasic plate. Then it is time for anaphase. During anaphase, the chromosomes break at centromere and the sister chromatids they move to the opposite ends of the cell. The last stage of karyokinesis is telophase, where the nuclear membrane reappears, the nucleoli reforms, and the chromosomes uncoil into chromatin network. Finally, after this, there is cytokinesis. Cytokinesis is the actual separation of the cell by the cytoplasmic division. At the end of it, we get two daughter cells formed from the single mother cell. The next could be a G0 phase. If the cell remains out of cell cycle, it may remain in a complete resting stage, which is also called the quiescent stage. During this time, there will be no division that takes place. But yes, there can be differentiation, specialization, de-differentiation, as well as redifferentiation, which is commonly seen for the meristematic tissues entering into the simple permanent tissues. But the cell can also proceed into another cell cycle if all the conditions are fulfilled for the cell to grow. Then the cell starts to repeat all the cellular events once again, and then it enters into another cell cycle, starting with growth through all the cyclic events. Now let's see what is the duration of a cell cycle. Actually, the duration of a cell cycle is not fixed. It is considerably different in different kinds of cells. For a typical rapidly proliferating human cell with a total cycle time of 24 hours, the G1 phase might last about 11 hours. The S phase around 8 hours, while the G2 is about 4 hours, but the M phase is approximately only around 1 hour. The other types of cells, however, can divide much more rapidly. The yeast cells can complete all the 4 stages of the cell cycle in just 90 minutes. The early embryonic cells take even less than 30 minutes to complete the entire cell cycle. There are no G1 or G2 phases for the early embryonic cells. There is only DNA replication with a short S phase that alternates with the M phases. In contrast to the rapid proliferation of embryonic cells, some cells in the adult animals cease division altogether, like the neurons in your body. Many other cells divide only occasionally when needed to replace the injured or the dead cells. They include the hepatocytes of the liver, the cells of the lungs and the kidneys, and even that of the skin fibroblasts. These cells exit the G1 to enter G0 or the quiescent stage, where they remain metabolically active but no longer proliferates unless approached extracellularly with signals that are generated to resume the cycle. Now let's try to see how was the concept of cell cycle born. So the discovery of cell cycle was done by Alma Rose Howard and Pelk. They were the first to ascribe a time frame to the cellular life, as they proposed the existence of 4 periods in the cell cycle. During that time, they called them as a period of division, a pre-synthetic G1 phase, the S phase, and a G2 or the pre-mitotic period. This was all that it started with right in the beginning of the cell cycle. Thank you for watching. In my next video, we will discuss about the quality control checks of cell cycle. I hope you liked my video. So I'm expecting a thumbs up. Please do like, share and subscribe if you find it valuable for you. Thank you for watching once again.